I decided that this time we're actually going to start with the short answer question because we ran out of time for our short answer questions that we had planned for 2 weeks ago. So we're going to start with the short answer question, then go into multiple choice. Then moving forward, I'm going to pick a full short answer question, like we've got a full multiple choice question paper, and then we will work through those two papers simultaneously. So 15 minutes on short answer, 15 minutes on multiple choice for each time that we do past paper questions. And then we will just deal with whatever topics we get that week. So today we're continuing on. In the last lesson, we started this, it's summer 13, paper 22, question 4. Okay, and it was we were looking at biological molecules and carbohydrates in particular and figuring out different types of carbohydrates. And then we spent a bit of time on looking at a cell membranes question. So the background to this I kept here for us. It is that in some organisms, trehalose is used as an energy store and gives protection against the harmful effects of very low temperatures. So trehalose was one of the molecules we identified. Trehalose is sometimes referred to as a cryoprotectant, so protecting from freezing, allowing organisms to survive in freezing conditions. So freezing temperatures can damage the cell surface membrane and membranes within the cell. So first off, we considered what's the importance of the cell membrane, okay, and what does it do, so why do we need it? The question then goes on to say freezing temperatures can also completely stop enzyme activity by causing the molecules to undergo cold denaturation. Now, this is not something that is included in your syllabus. It's not something that you study. So it's important that you use the information that they're giving you in the question and use your understanding of enzymes and protein structure and denaturation and apply your understanding into the situation. So they tell us enzyme activity is not recovered when temperatures are increased to a normal working temperature range. So it is exactly the same as any kind of other denaturation. Okay? If it's not recovered, again, we don't have bonds reforming. Okay? In order to build an enzyme, we have to go through primary, secondary, tertiary structure. We can't just poof, there's an enzyme. It can't just reform itself. So as soon as we denature an enzyme, it's gone, it's finished. Okay, so first off, they say to us, explain the mode of action of enzymes. So they don't actually ask us anything about this cold denaturation to start. Part 2 is only where they say, suggest how the molecular structure of the enzyme changes during cold denaturation. And what they're doing is they're actually leading you into your understanding of the question. They're being really nice to you. Because if you think about the mode of action of enzymes, that's going to link to things that happen in denaturation. So what are the bullet points that we want to bear in mind when we're talking about mode of action of enzymes? There are a few, and they have all those technical terms in them. So the first thing you want to say when we're talking about mode of action, how an enzyme gets its job done, is that the active site has a specific shape. Okay. You want to also point out that it's not just that shape is like a specific shape. It is complementary to the substrate. Okay, remember when we talk about enzymes, we use complementary with an E in the middle because we're talking about shapes that complement each other, not people saying nice things about other people. Okay, so in here we can split into two different things. Remember when we talk about complementary and specific shape, we can mention lock and key. Okay, so this one is a precise fit. And we can also mention induced fit. So this induced fit is where you have your slight alteration. To even further improve that fit between the enzyme and the access, to further improve the fit between the enzyme's active site and the substrate. So we have an even better catalysis of the reaction. Okay, so what else do we need to talk about? So we know that these things are complementary, they are specific. What's going to happen is that the substrate is going to enter the active site and it will be held there. Because it can't just sort of randomly bump in and out. You're going to have that involvement of hydrogen bonds between R groups of the amino acids in the active site and portions of that substrate molecule. So, hydrogen bonds between R groups of active site and the substrate itself. Okay, this then, remember, as soon as we have bonding, we form something different. We no longer have an enzyme and a substrate. Now we've bonded them together, so it needs a new name. So this forms an enzyme substrate complex. What is the point of doing that? How do we now actually do the job of catalyzing this reaction? What we're doing is we are lowering the activation energy. So activation energy is reduced. And if you look at this, we've got 1, 2, 3, 4, 5 solid bullet points and 2 subpoints, and it's only 3 marks. So we've definitely covered our bases in terms of a thorough understanding of the mode of action of enzymes. And that's what you want to do when you're revising, is you want to aim for that full thorough understanding, even though it doesn't really fit here, okay? Because then when you get into the exam, you can pull out the bits that are particularly relevant, or worst case scenario, the bits that you can remember. If you can only remember some of it, at least you've remembered more, you can still remember your 3 marks. So mode of action of enzymes, we'll turn this into some beautiful points that would link onto one another and have your full answer. Okay, but it's all about that shape, being specific, being complementary, forming that enzyme substrate complex and reducing the activation energy. And that is the mode of action of enzymes. That's how they get their job done. It's how they catalyze reactions. So now that we've focused on all of that importance of shape, let's move on to part 2. Suggest how the molecular structure of the enzyme changes during cold denaturation. Well, we know that denaturation of any kind is going to affect bonding. Okay? So the bonds in the molecule are no longer going to be working as they should be, so we're going to lose that shape. If we go back into the information in the question, they actually told us that enzyme activity is not recovered. So we're not getting the shape back when the shape is gone. So it is exactly the same. So first thing is that we have loss of tertiary structure. We can even be more detailed here because we know that many enzymes have a globular structure. Okay, we're going to lose that. Okay, because we've lost bonding. Okay, we don't know which bonds are going to be affected by cold. So let's not get into that. But let's think about if we lose that tertiary structure, the most important thing that we lose is that active site. So the active site changes shape. And it's no longer complementary to the substrate. Okay, and that's what's going to happen. It's the molecular structure of the enzyme changes. We're losing the tertiary structure. We're losing the active site. It's changing shape. Is no longer complementary. So we've described now what is the effect of that cold denaturation on the protein structure. Okay, so we've dealt with part D. We're happy now with our freezing temperatures and our cold denaturation. And I just want to point out again, the bulk of the marks from this question is things you know. Then there's a little bit of application in for a suggest, but it's using the information that you most definitely know, plus the information that's there in the question. They're not going to give you questions that are out of left field. Okay? There's always going to be something there to help you. Next part of this question is one of those types that often trips people up. So cryoprotectants such as tree halos are of particular interest in their application to preserving cells, tissues, or organisms for future use. Okay, that makes sense. We can freeze them, but they'll stay intact. An investigation was carried out to find a protective effect given by different concentrations of two cryoprotectants, tree halos and glycerol, on a respiratory enzyme. Okay. The enzyme was subjected to a freezing temperature and then returned to its optimum temperature. The activity of the enzyme was measured at its optimum temperature. Okay, so under the best conditions of these enzymes, what happens after they've been frozen and then returned? So 4.2 is a graph showing the results of the investigation. So what we've got at the bottom here on the horizontal axis is cryoprotectant concentration, millimole. So how much protectant they put in. And then on our vertical axis, we've got percentage of maximum activity. So they know the activity that we should have at optimum temperature, that would be 100%. So that's like an unaffected enzyme. And then all the way down to 0, meaning that that enzyme didn't get any functionality back at all. Then we've got two lines. We've got one for tree halos with our X's, and we've got one for glycerol with these funny dots. Okay, makes sense. So when there is no cryoprotectant, there is no activity. So they're reiterating that point from the beginning, that when we have cold denaturation, then the enzyme activity is lost. So even just with a little bit of protectant, then we get some of that activity back. Tree halos looks much better. It's going up much a steeper gradient of this curve. We get a lot more activity back with lower concentrations. Whereas with glycerol, we've got to add quite a lot of glycerol in order to get back that functionality. Okay, always take the time to understand your graph properly. So next bit, with reference to figure 4.2, describe the results of the investigation. See how they even put the describe in bold. Describe the results of the investigation. Not describe and explain, just describe, which means it went up, it stayed flat, it was 0, it was maximum, this was higher than that one, this was lower than that one. That's all the sorts of things that you're doing, okay? Just describe it. So what do we have if we think about descriptions here? Okay, well, first off, we want to look at the general overall pattern, and they both increase. Increasing cryoprotectant increases percentage of maximum activity. So, for both, again, we're going to go for minimal bullet points to maximize information here. They're not necessarily in the same style that you would write in an exam. So both increasing concentration increases activity recovered. Okay, so that's that general going up. Okay, so now let's look at the comparisons. Okay, so we say tree halos gets all the way up to max, okay, here already. It's got a steeper increase in the first 10 millimoles. But where do they come together? Okay, so they come together about here. Okay, so what is that point? That is 90. So up to 90 millimoles, tree halos is more effective than glycerol. So up to 90 millimoles, tree halos leads to higher max activity. So higher percentage maximum activity, we would write in full in our exam. Okay, at the moment, we're just taking notes. Let's give ourselves a bit more space. Okay, so now let's consider this big difference here at the beginning, because you see how glycerol it increases here from 10, but from 0 to 10, there's this massive difference because tree halos goes up to 80, whereas glycerol only goes up to like just above 10. Okay, so that is a description. I mean, it's an important description that at these low concentrations, tree halos is much more effective. So we could say that tree halos, steeper increase at low concentrations. And here is an ideal thing to use our data. So it's 0 to 10 millimoles. Tree halos is going to go from 0 to 80%. Glycerol is going to go from 0 to 10%. That is a massive difference. Okay, so a nice little point to describe. Because whenever we're describing a curve, we're sort of looking at the ups, the flats, the downs, the fluctuations. Whenever we're comparing two things, we want to look, where are they the same, like we did at the beginning, they both increase. Where are they different, like this, we have steep versus shallow. So going back now, we've looked at the beginning, okay, then they sort of roughly go more similar. But the thing is that they both get to max, they both get to 100%. So at higher concentrations, they can both produce 100% of maximum activity. And again, here's our ideal. We said this one here is at 30. So tree halos is at 30 millimoles. Glycerol was up here at 90 millimoles. Okay, so that is then, we've talked about the maximums, we talked about the increase, we've talked about the general trends. Up to 90 millimoles, tree halos is the max activity. Okay, yeah, so, and we're on 4 marks. Now we've used 1, 2, 3, 4 bullet points. We've got supplementary figures for 2 of them. So we actually do have space to turn these 4 bullet points into something that's a more substantial sentence or a more substantial point, or actually has tree halos and glycerol in instead of T and G. And that's the idea, that your answer, when written properly, should fill the space that you have available. Depending on your handwriting size, it can vary. That's what you're aiming for. So how would you do this in an exam is one of the things, I mean, you can't write it out like this and then turn it into sentences. You've only got one space to write on. But whenever you have a graph, there is nothing stopping you doing a similar thing around the graph. So in our sort of notes that we've put here, you can add in your points on here, that they're both going up. And so you'll probably get to some sort of version of a shorthand which you use for your own personal understanding of graphs. And then you can turn that into nice sentences for your examiner. You can scribble to your heart's content on these graphs, because what's being marked, what's being analyzed is the answers that you write just on those dotted lines for the marking. So make sure that you take the time to really understand what's going on in the graph. Okay, have we got any questions relating to these short answer questions, or are we happy to move on to multiple choices? So it's the same paper that we were working on last time. We're going to keep going with this paper all the way to the end. So May, June 2018, paper 11. We are kicking off now with question 5. So we have a diagram of a cell, and we should recognize quite a few things inside this cell. We have various different things. Let's change color and have some fun. So radioactively labeled nucleotides are introduced into a cell. What have we got here? So 1 Lines with dots, we have rough ER. 2, great big circular thing surrounded by rough ER. That will be our nucleus. 3 is a mitochondrion. It's only one of them is labeled, so it's a mitochondrion for 1. And 4, the one that looks a bit like a Wi-Fi, there's our Golgi apparatus. Okay, so nucleotides. In which cell structures will the radioactivity first become concentrated? So where are we going to be using these radioactively labeled nucleotides in our cell? I'll give you a moment to think. This is a slightly unusual question. Okay, so normally this question is about amino acids and it's all about proteins. But this question says nucleotides. We use nucleotides to build DNA and we use nucleotides to build RNA. Okay, so where do we find DNA and RNA? Most certainly in the nucleus and also in the mitochondrion. Because remember, your mitochondrion has that circular loop of DNA. So we are looking for 2 and 3, which is our answer C. So the rough ER and the Golgi apparatus are there for the question that is about amino acids, not about nucleotides. So make sure that you are always reading very carefully because one word in a multiple choice question can knock you out completely. Okie dokie, next question. What is the general formula for amylose? Amylose is alpha glucose. Agree? So alpha glucose is C6H12O6. However, ladies and gentlemen, that changes, okay? Because remember that we have turned these into ring formulas and we have joined them together. So when we talk about the general formula for amylose, we are not just talking about lots of alpha glucoses as they are, because in order to join things together, in order to make rings and join them together, we've got to make a plan to share electrons, in which case we need to remove atoms. So C6H12O6, these can't be joined together. What do we move? Do we remove hydrogens, oxygens, carbons? Well, if you think about it, you've got carbon 1, 2, 3, 4, 5, 6. And you're never going to remove that carbon. That carbon is the skeleton of the molecule. So anything with a C5 is out. And that leaves us with C6H10O5. So think about that. You've lost an H2O. And now you should be smiling broadly because you know that a condensation reaction removes H2O. So now you are happy with your answer, that the general formula for amylose is C6H10O5N. Even though it's not something you specifically studied, you can use the information that you've learned to figure out the question. Question 7. Now we're talking about molecules again. The diagram shows relationships between some important molecules and bonds. Okay, that makes sense. So 1, 2, and 3, we have 3 circles. Okay, we have peptide, ester, and glycosidic. We've got cellulose and amylopectin. We've got triglycerides and vegetable oil. Okay, so in 3, these guys are lipids, and this is the bond for a lipid. Okay, makes sense? Here, cellulose, amylopectin, both of these are carbohydrates. Glycosidic is the bond for carbohydrates. Peptide, ester, glycosidic, these are all bonds. You can spell bonds, it helps. Okay, now remember and make note that I've done all of this before I've even looked at what's coming next. I want to make sure that I understand the information that I'm given in the question. What is represented by circles numbered 1, 2, and 3? And then we've got lots of different options in here. We've got lipids and carbohydrates, and then we've got bonds formed by condensation and bonds formed by hydrolysis. And we're thinking, no, that's a silly question. Bonds are formed by condensation. Bonds are broken by hydrolysis. So we can add here condensation. And now we know we're looking for 1 is bonds formed by condensation. Number 2 is carbohydrates. And number 3 is lipids. And that leaves us with option A. Straightforward, straight into it, without getting distracted or confused by the options within the table. Answer the question first, then look for your answer. We've got time for one more question today. In unsaturated lipid molecules, where are double bonds located? Unsaturated. Okay, so what is a lipid molecule? We have our glycerol. We have our fatty acids. Okay, let's just do it for a triglyceride, why not? Okay, so we have 3 fatty acids. Now, unsaturated means that we have a kink in our chain because we have this carbon to carbon because we don't have all the hydrogen. We're not saturated with hydrogen, we are unsaturated, okay? So this carbon to carbon is a double bond. Okay, so we're happy that we have double bonds in our fatty acids. So D is out. Okay, so do we have double bonds in glycerol? So think nicely about glycerol. And glycerol is just a whole lot of carbons and H's and OH's. There's no double bonds there. Okay, so we don't have within glycerol. So B is out. Then between fatty acids and glycerol, this is our ester bond. Formed by condensation, broken by hydrolysis, exactly the same as basically any other bond that we form. And there is definitely no double bond involved in that carbon-carbon with the ester bond. So it's not A, which leaves us C. We are confirmed, we are sure that the double bond in unsaturated lipid molecules is just within that fatty acid molecule, where you have that carbon-to-carbon double bond, which gives us the kink, which changes the whole fluidity of that lipid molecule. So that has taken us to time for today. So moving forward with past paper questions, we will do this, keep going with this every second week, random topics.